All right, this is the first chapter of the second unit, and the topics that we're going to be covering in this unit are primarily about microbial growth, and by growth, we're actually talking about replication here, not a size increase, and then how we control their growth, and we control them in a variety of ways, like common chemical disinfection, all the way to really sophisticated antimicrobial drugs. And then we're going to switch gears and just kind of introduce ourselves to some key vocabulary about pathogenicity and epidemiology, which we will put to good use in the last unit of this class, which is all microbial diseases. So let's just get started with the mechanics and the requirements of microbial growth. And I will say, this doesn't really seem like a cutting edge chapter, but it's actually something that's getting more and more important to understand, like the very basic growth requirements of microbes as we are seeing some of our drugs lose their efficacy. We're actually going back and looking at these like characteristics, and it's actually really helpful in medicine. So it might seem a little bit boring, but it is still incredibly useful. First thing, from our first unit, you got a little bit of an introduction about all of the variety and the diversity of different types of organisms that fit into the microbial world. Remember that bacteria in particular are incredibly diverse. What they need to live, where they live, how long they live, what they need to replicate is just all over the place. And an understanding of that growth phenomenon actually helps us identify them in some cases. Sometimes we look at maybe byproducts that they produce as they grow. We can test for that in a lab or some kind of, newfangled science thing. It's important information. It's good to know that we're going to be working with pure cultures as we allow for microbes to grow. Remember, if it's a mixed culture, you're going to get all sorts of mixed information and you don't really know anything about it. Since you can't really see it, contamination is a big source of concern for us. And I'll be talking a little bit about some of the methods that we use to ensure pure culture in this chapter. This percentage, 1%, is actually something that I really want you to process and ponder. The fact that we can, in today's scientific world, only grow 1% of microorganisms in pure culture is mind-blowing. Can you imagine if we could only touch 1% of the types of rocks on the planet or chemicals? I mean, the way that microbiology is in its infancy is probably determined by how limited we are in how we know to give microbes what they need to grow. We just can't grow them. We might see traces of them, we might see like little pieces of their DNA and know that they're out there, but this is one of the most significant limitations in our science. Okay, there's a few characteristics we're going to go through and see how it affects or influences microbial growth, and that's going to include temperature, pH, osmotic pressure, which is solute or chemical concentration, and then these very specific chemical requirements like why do they need sulfur, why do they need phosphorus, that stuff. We're going to start with temperature, and temperature will basically be a bell curve. There's going to be a temperature that's too cold for them to thrive or grow. Below that, they're just like nothing. They're super happiest at the optimum. That's where we're going to get the fastest rate of growth. And then they get hot at some maximum temperature and also stresses them out. And that will be above that temperature, nothing's going to happen. Here's what those curves look like. We can basically just inoculate a bunch of different petri dishes, throw them in different incubators that are at different temperatures, and then kind of record how well things grow. So you'll see that we have these four kind of different colored curves. Psychro is a prefix that means cold. Phile is a suffix that means lover. So the first little curve that's purple on the left are psychrophiles. These are the cold-loving bacteria. These would live somewhere, like maybe in nature, like in the snow, or maybe even in the permafrost. These could live in your refrigerator because your refrigerator is quite cold. The temperatures here are in Celsius, so it's actually not as not as Fahrenheit as it looked, but there's definitely a range of things that could cause food spoilage that you could come into contact with. Mesophiles is next. Meso means middle. These are microbes that love it in the middle, and their optimum is going to be 37 degrees Celsius, which is human body temperature. These are not only the most common, but the most interesting to us because they're potentially human pathogens. Thermo and hyperthermo are the very hot-loving microbes, and these are going to be all over the place. Everything from decomposing vegetable matter like compost. If you've ever seen steam rising off compost or bark chips or something, those are definitely from these thermophiles. And then the hyperthermophiles usually love it in very extreme environments that are extra, extra hot, hotter than boiling water. So I've kind of created a range for you. I do think it's good to know that there are psychrophiles and trophs. If you see the difference between a phile and a troph, trophs means tolerant, and these are considered a subcategory of that group, and they are just slightly above the range that the psychrophiles would be. Mesophiles, like I said, are really important medically because of that optimum 37 degrees Celsius temperature. And then the thermophiles, these are really interesting. They're environmental microbes, and some of them live in really, really cool environments. So here's a picture of watermelon snow. If you've ever seen watermelon snow, it can almost look like there's blood or something on the ice. This is a microbial bloom. These are definitely cold lovers, but I would just say never, ever, ever try to melt colored snow, any color. This can actually be a little bit toxic. This is a thermal vent. It's a structure that we refer to as a black smoker. You can see that it's just bilging and billowing black smoke and soot and chemicals and all sorts of stuff. This is a volcanic vent at the very bottom of the ocean. It's under a lot of pressure. There's a extremely high temperature here. It's basically like as hot as lava. We used to think nothing could survive there, but we know that there are these like extreme hyperthermophiles that thrive there. And I'm just going to give a shameless plug for the microbiology program at Portland State University. They are exploring this particular ecosystem. They have 2 submarines. Some of these smokers are like off the coast of South Africa. And if you ever want to take a higher level micro class, it's very, very interesting stuff. A lot of new discovery is happening there every day. And I just think this is a really interesting phenomenon. These black smokers are actually more diverse in terms of life than the rainforest, which is just, it's like staggeringly, mind-blowingly cool. Okay, I just wanted to mention why those minimum and maximum temperatures matter. The minimum and the maximum temperatures are usually where we start to see membranes like lose their structural integrity. Either they freeze up or they get too thick and kind of like maybe solid. Those temperatures are not necessarily survivable. Also, the enzymes are temperature dependent. So think of like hard boiling an egg. That protein in the egg white gets seized up and it becomes like a solid and it's not going to function anymore. That's what's happening here. We're freezing, or we're basically cooking those cell structures to the point of no return. pH is next. pH is acid or base in a solution. You should know most bacteria like it kind of neutral. I will just point out that your blood is within that neutral range. It's on average 7.4 in pH. So usually microbes are going to be fine inside that pH range that's fairly neutral. There are some exceptions. There are some microbes that might like it a little more acidic. There are some acidophiles, which are acid lovers, that can grow in lower pH ranges, and that can be down to pH 2. 2. So pH 2 is the maximum like acidity of your stomach, and they are like completely happy there. So occasionally, like the pH is an interesting thing to know. In this really cool study that I read several years ago, they were integrating pH detection in bandages. And I think this is so brilliant because a lot of times you're not aware that you have an infection until it's really bad. So if we had like little pH detectors like, hey, something is shifting acidic, or even had like specific identification like antibody products like integrated or incorporated into that bandage, it would be so cool, especially for people who are at high risk of infection, such as after a surgery, or if someone has like out of control blood sugar or had an amputation, or just sheets for someone who's medically fragile. Like if we could have like a detector built in capitalizing on the perfect environment for these pathogens, or maybe acidic byproducts that are being produced as they grow in that environment, I just think that'd be so smart. Anyway, this hasn't really made it to the US yet, but I'm waiting every day. All right, osmotic pressure is our next growth characteristic, and this is basically a concentration of sugar or salt. By definition, osmotic pressure is just how salty or sugary a solution is. And I will say that it's not likely to run into a lot of high sugar environments for microbes other than things like ripe fruit or whatever. And usually that ripe fruit doesn't last very long because the microbes just like eat it all up. But salt is something that is fairly typical in nature. We have the salt in the ocean. We have salt in like these kind of less free water systems like the Great Salt Lake. There's salt crystals inside the earth. There's all sorts of like environments where salt is actually natural, and usually the saltiness is in high concentration, so it's a hypertonic environment. And when that does is it causes cells to basically burst. These come in either a osmotic required for life and growth. Those are the obligate halophiles. Halo is a term, by the way, that means square. And I always remember that salt crystals are square. So an obligate halophile requires like a lot of salt, basically. Facultative is usually like, well, it could maybe have a preference, but it doesn't necessarily require it. So tolerant is a better word to associate with facultative. We have kind of both levels of those. Nitrogen, sulfur, and phosphorus, carbon, those are also molecules that you've probably heard about in other classes. I will say carbon doesn't really have a special like application here. Carbon makes a lot of bonds that are easily broken. Microbes need carbon for energy. Period. Same reason you need carbon. So just know that sometimes they can produce their own carbon energy sources, kind of like a plant can. And it just sort of depends. But I would say if you just associate the word energy with carbon, no matter how they manufacture that energy from it, you're in good shape. Next we have nitrogen, sulfur, and phosphorus. Nitrogen and sulfur are ingredients of amino acids, and amino acids are the subunit of proteins. So we know that microbes need proteins to be enzymes to do different chemical pathways. Same thing here. You need nitrogen and sulfur for amino acids. They need nitrogen, sulfur for amino acids. The other one, phosphorus. Phosphorus is a key ingredient of the genetic material, DNA, RNA. It's also an important piece of ATP, which is kind of an energy currency molecule, and also the phospholipid membrane. So I would associate phosphorus with more than one thing: genetic material, ATP, and phospholipid membrane. I will also just point out that there has never been an organism that we've discovered on the planet Earth that can survive without carbon, nitrogen, sulfur, and phosphorus. So we kind of think these are like the most essential ingredients that any organism needs, not just a microbe, but particularly microbes, because they're fairly minimal, still require all of those chemicals. Trace elements are things that they need in really small amounts, and these are going to be really weird things like boron or iron, or I don't know, they're kind of all over the place. They're just really weird exotic things that they need for some specific reason. We need iron, for example, because it is the cofactor of hemoglobin. It allows hemoglobin to actually bind to oxygen in your blood. That's the only real reason you need iron. They'll need little things like that too for little cool processes here and there. I would say if you look at the ingredients of a vitamin, like all of the different cobalt and, just random stuff, it's going to be for the same thing. So I would think of trace elements as just kind of exotic, very small amounts required for some particularly special pathway. Next, we have oxygen, and oxygen is important. Oxygen is usually described as either being aerobic or anaerobic. Aerobic means with oxygen, anaerobic means without. And we're going to see a little bit more of a range here. We're going to have obligate aerobes, and then we're going to have obligate anaerobes. That's basically black and white. Either you need it or you cannot survive in the presence of oxygen. We will also see some that are a little bit in between. I think it makes sense with some of these vocabulary words if you take apart their names. We'll have some microaerophiles. Those need oxygen in kind of a small or microscopic amount. Aerotolerant anaerobes, they only replicate with one environment, but they can survive in both, which is kind of a little bit nuanced. Facultative anaerobes, those can kind of swing both ways, but they usually only kind of tolerate an anaerobic environment. These are like really important vocabulary words, and I will say there's actually only one structure in the human body that is obligated to have oxygen, and that is the mitochondria. So if you think about it, it's kind of cool. I know not all of you love the endosymbiotic or endosymbiont theory, but it's pretty extraordinary that you have so many body systems that service that little dude. I just think that's cool. All right. Next, for oxygen, I want you to be able to visualize what those oxygen tolerances are. And if you're looking at a broth, like a liquid culture of bacteria, where the bacteria are growing can actually tell you. If it's close to the surface, it's usually something that wants oxygen. If it's as far from the surface as possible, it is going to be more anaerobic. And then we'll see kind of variations of those based on their tolerances in different ways, if they're facultative anaerobes or aerotolerant or microaerophiles, etc. It's actually really cool. You can develop a certain sense of their personality just based on checking it out in a broth culture and seeing where that blob of bacteria are. Let's talk more about growth. So, we need to prepare the recipe of their nutrients in a sterile way. That's why we have our autoclave. We're going to basically put all these weird ingredients together and sterilize it, and then we're going to inoculate it, which means we're going to introduce the microbes into the medium, and hopefully we get some growth there in the form of a culture. Usually in a lab, it's much easier to have it grow on top of a medium, but depending on the oxygen tolerance, we might inject it or, create a little pocket for it so that the oxygen is not a problem. For solid culture media, we use that agar stuff, which is a complex polysaccharide, like I said, from seaweed. I mean, you could honestly do this at home. Like, you could go to the coast and get some seaweed and then, probably boil it and extract the agar. And then you could probably microwave it or something to sterilize it, and you would have a growth medium. It's ideal because it is not metabolized, right? And we know exactly every single ingredient that goes into a growth medium. We have to, because we want to know if it's something that microbe needs. There's times when we put really complicated biological molecules in it, and I will say it's really hard to chemically define everything that's in meat or everything that's in plants, but we usually put stuff that's complex into agar if we want to get something special to grow a little bit better. I will say agar itself can be antimicrobial. That's a problem, but just know that we still use it as a standard for pretty much 100% of basic microbiology growth. If you see the word enrichment media, it should sound like it's something extra in there, and this is for stuff that's going to be harder to grow normally. We'll give it something, I don't know, special. We might even give it something that inhibits other stuff from growing so that it has more room and a little bit more of an advantage. That can be really helpful if you're just trying to grow something rare and, I don't know, just a little bit more special needs. Okay, how do we get pure cultures and know that we're working with a pure culture and then measure the actual growth is our next topic. First, we do a very simple procedure for spreading bacteria apart on a plate called a streak plate. This will allow the individual bacteria to be separated in a way that they will only grow without contact with any other colony, and you can just pretty much assume that it's clones that are derived from that original source, which makes it quote-unquote pure. We have two ways of measuring growth, and I'll look at a streak plate first and then go through those direct microscopic counts and serial dilutions. This is a glowing, beautiful streak plate, and how it works is that you take your loop and you spread it back and forth, kind of just diluting your bacteria as you go back and forth. You never backtrack, but it just kind of physically spreads those bacteria apart. And you can see that there are some that are touching. Those are not considered pure. But the colonies that are physically distant enough from one another that they're growing just a perfect little round colony, those would be considered pure. That would be something that we could sample, run tests on, stain. We would only grab from one of those pure colonies. Streak plates are nice because they're really fast and cheap to do. Now, let's talk about how do we count how many bacteria there are. This can be a painful process. You know that these are very small. So what you would want to do is take a sample. You put this really fancy like cover slip on this fancy slide with like a laser etched microscopic grid so you don't lose count. And you actually just physically count how many little individual bacteria you see on this slide. It might seem silly to do this, but this is actually how we will measure how many microbes create an infectious dose. So infectious doses are required if we want to tell someone what their risk is for maybe being exposed, or if we want to say, oh, well, this N95 mask like will filter this many viruses or bacteria out of the air. Someone actually has to physically count it and test it, and this is usually the method that we would use. How about a serial dilution? Serial dilution is nice because it's, again, not as tedious as like counting under the microscope. And here's how it works. First, what we want to do is know how many bacteria are present in a sample, but we don't want to just count everything in a one big old gallon jug or whatever that is. What we want to do is take a 1 ml sample from that jug and put it in 9 ml of sterile broth. We want to dilute it to 10% of its original amount, which would be probably too many to count still. So you can keep doing that. You can take a small sample of that dilution, dilute it again. If you did it a second time, it would be 1/100th of the original concentration, and then again to 1/1000th, 1/10,000th, 1/100,000th. Basically, you want to dilute it out so there's only a few bacteria in there, and then you can put that on a Petri plate and see how many colonies that grew. Each colony would have represented a single bacteria, and then you can just multiply how many times you diluted it, and that will basically give you an estimate of how many were in that original source. This can be very useful. Is it exact? No, it's not. It's going to be a round number, but it sure saves you a ton of time from having to like count to like a million. Microbial growth follows a typical curve pattern, and this is actually a really important part of this chapter. I want you to really look at this growth curve because it actually changes over time. When you create your culture at first, there's a little bit of a lag, and that's just like where they're shocked. They're in a new place. They're like, whoa, what just happened? They're not creating any division at that point. They're just getting ready. So no new individuals are being born. It's just getting adjusted to their new environment. And then they're going to start to replicate, and that's going to be exponential growth. That is going to be rapid duplication. Duplication could be anywhere between 10 minutes or maybe 10 weeks. It just depends on the species, but they are replicating exponentially. Then they're going to start to run out of resources. Some of the originators are going to get old and die, and what we see is that the population stays stationary. That's where the birth is going to equal the death, and it's not necessarily growing in numbers. It's just kind of stable, and then they're going to start to die. They're going to run out of resources, they're going to run out of food, they're going to have too much like competition. We're going to see that they'll die off, and that can actually kind of have a steep and a less steep portion. So here's my curve. It kind of looks like a little bell curve. The first is the lag phase where the microbes are introduced and they're not growing yet, they're just kind of chilling out. And then they're gearing up for that logarithmic or exponential phase of growth where it's like rapidly increasing in the number of bacterial cells. And then we'll get a kind of a flat stationary phase where birth equals death, and then they're going to start to die. And like I said, the slope of that death can vary. It's going to start out pretty fast and then it might slow down a little bit. It just sort of depends on the species. Now, I've mentioned reproduction a couple of different times, and usually what I'm talking about is binary fission in relation to bacteria. But there are some weird other ways of reproduction to occur in the microbial world. It could be a bud, especially if it's like a algae or maybe even a freshwater protozoa. They can bud sometimes. They're so weird. There can be conidiospores, which are a type of like fungus. There can be fragments of filaments that can kind of just break apart and then they can basically keep going as two. There's other random ways, but binary fission is the process that we talk about the most, and it is relative to bacteria. The nice thing about bacterial binary fission is it's super easy. There's no steps like there is in mitosis or something. It's literally just duplicate everything identically and then split apart. I often refer to this as copy and paste. If it's a perfect copy and paste, it's a clone, right? And there really aren't any more steps than that. It just like duplicates everything and then boom. Couple other things that I wanted to mention about growth. We talked a little bit about the glycocalyx in the previous unit, that kind of sticky outer carbohydrate surface. A biofilm is similar, but it's important to note that this is not an individual structure. This is a community structure. A biofilm is like a really heavy encasement of slime that covers an entire area. If you go to the river and the rocks are kind of slimy and slippery, that's a biofilm. If you are interested in dental hygiene, your dental plaque or your dental tartar, which is calcified plaque, is a biofilm. There are more than one species that can be inside of a biofilm, and we associate with biofilms with potential pathogenicity. And the reason for that is it's really hard to get chemicals or drugs to penetrate through that biofilm. So particularly if there is some kind of instrumentation going into a person's body, like a catheter or, maybe a PICC line or something, the biofilm accumulation can actually be extremely dangerous. So think of a biofilm as part of a community of microbes that are growing together, that it just protects itself with this really thick polysaccharide slime. Here's what a biofilm looks like. This is an SEM, right? Like you can see that there's lots of stickiness in there. It's just going to be really protective, and to me it just looks really sticky and slimy, and I don't know, it kind of looks dangerous. All right, last thing, storing pure cultures. We got to be able to store them, and we can do that in a couple ways. We can just kind of let them hang out in the lab where they'll probably dry out in a couple of weeks. That would happen on a petri dish or a slant. That's not always ideal for long term. For long term, what we want to do is take the water out of it, and we can either do that by putting it in glycerol to kind of replace the water and then freeze it really cold, like minus 70 degrees Celsius, or we can dehydrate it, which is a process called lyophilization. If you've ever had like a cake mix or like something that you just have to rehydrate, that's what lyophilization produces. It's like a freeze-dried powder, and it could have like milk and all sorts of eggs or weird stuff in there. But by removing the water, all those microbes are basically unable to metabolize, and so it's shelf stable. It's really awesome. You can store it for a really long time. So it just depends on what you need, but you do have to be able to store this stuff so that we can use it later.